Charleston, South Carolina. October 2013, a construction crew is digging a routine utility trench on Anson Street, 6 inches below the asphalt of a parking lot in one of the wealthiest zip codes in the American South. The backhoe stops, the operator climbs down, and what he finds in the dirt beneath that parking lot is about to rewrite what scientists thought they knew about the origins of an entire people. 36 sets of human bones. They dated to the second half of the 18th century, all of African descent, most believed to have been enslaved. Charleston's port had processed an estimated 175,000 Africans through its docks, roughly 40% of every enslaved person who entered British North America arrived through this one city. These 36 people had no names on any record, no grave markers, no documentation of any kind. The city had built a parking lot over them and gone about its business for 200 years. But here's the thing about bones. They remember what history forgets. In 2023, a team led by Dr. Raquel Flesks at the University of Connecticut, working with Dr. Theodore Schur at the University of Pennsylvania and the Anson Street African Burial Ground Project, published results that stopped the entire field. They had extracted ancient DNA from those remains, 18 full low-coverage genomes, 31 maternal lineages. It became the largest dataset of ancient genomes ever recovered from African-descended people anywhere on the North American continent. 36 people buried without ceremony, without names, without records. And science gave them something 400 years of history had deliberately refused to: origin. But what those genomes revealed was not the story that millions of people had been told. It was not the story the history books suggested. It was not the story that commercial ancestry tests had been selling. What those bones revealed was something older, more complicated, and far more important than anyone expected. Here's what this episode is actually about: a forced migration that reshaped an entire continent, tens of millions of living descendants. The agricultural backbone of the American South built entirely on the knowledge and labor of one specific group of people. And in the entire historical record, not one collective name, only a chromosome. That chromosome is still carrying information. And in 2025, researchers figured out exactly what it means. A chromosome is not neutral, neither is a story of survival. St. Helena Island, South Carolina, 1989. 15 Gullah speakers board a plane for Sierra Leone. They believe they are going home. When they land in Freetown and begin speaking, something extraordinary happens. The local Creole speakers understand them. The Gullah understand the Creole back. The moment is filmed. It becomes one of the most cited pieces of cultural evidence in the history of American identity, a linguistic handshake across 400 years and an entire ocean. For decades, that moment anchored the narrative. The Gullah came from Sierra Leone. The language proved it. The connection was real, and it was sacred, and it helped millions of people understand where they came from. Then the genome complicated everything. Dr. Kip Zimmerman at Wake Forest School of Medicine, working alongside Dr. David Reich at Harvard, ran whole genome analysis on Gullah individuals and compared them against 16 distinct Sierra Leonean ethnic populations. What they published in the American Journal of Physical Anthropology in 2021 could not be contained by the Sierra Leone origin story. Yes, the Gullah showed slightly higher genetic relatedness to Sierra Leone than urban African Americans did. But the key word is slightly. They were equally related to populations from Senegambia, the Gold Coast, the Windward Coast, West Central Africa, present-day Angola, and the Congo. The genome was not pointing at one homeland. It was pointing at a dozen. The slave trade records for Charleston confirmed exactly why. Of the enslaved Africans brought through Charleston's port, approximately 39% came from West Central Africa, Angola, and the Congo, 20% from Senegambia, 17% from the Windward Coast, 13% from the Gold Coast, only 6% from Sierra Leone itself. The origin story had always been downstream of the cargo manifests, and yet the language connection was completely real. That is the part that requires careful thought. When you destroy a people's language and force them to rebuild one from fragments, those fragments naturally cluster around whichever ports received the most ships during a particular decade. The Gullah language preserved the linguistic evidence of one dominant trade route. The body preserved every route. The Sierra Leone story was not wrong. It was the story that survived because it had to. Because when slavery strips away every anchor of identity, land, name, family, language, people construct the homecoming they need from whatever materials remain. The genome does not dishonor that. It shows how much more there was beneath it, and that how much more is precisely what makes the next finding so significant. To understand why the Gullah genome survived, when so much else was destroyed, you need to understand something about a parasite. South Carolina's Rice Coast, Summer 1733, a European plantation overseer collapses in a tidal swamp. By August, white mortality on the Sea Islands during the rice growing season is running high enough that wealthy planters have adopted a systematic practice they call absenteeism. They leave. They flee inland. They flee north. They disappear for months at a time to escape what colonial records called the country fever. They leave behind the people who built the fields, planted the seed, and managed the tidal irrigation systems, because those people, unlike the planters, do not collapse. The parasite was Plasmodium falciparum malaria. West and Central Africans carried partial genetic resistance to it through two specific mechanisms, the sickle cell trait and Duffy negativity, adaptations built by millennia of living alongside this exact parasite in the same river basins and coastal marshes where they had farmed for generations. Europeans had no such adaptations. The diseased geography of the Sea Islands made them effectively uninhabitable for European supervisors during the entire agricultural season, which happened to be exactly when the rice had to be grown. The parasite that killed European colonizers in the 1730s is the direct reason Gullah Geechee. Mitochondrial DNA haplogroups survived intact long enough for a researcher at Wake Forest to sequence them in 2021. That is not a metaphor. That is a causal chain. By 1708, black inhabitants outnumbered white inhabitants in South Carolina, the only British North American colony where this was true. A European visitor to Charleston in the 1730s described it as looking, in his words, more like a Negro country than a country settled by white people. The plantation task system, which assigned enslaved workers a defined daily task and allowed limited autonomous time once it was complete, developed partly because there were not enough European overseers present during fever season to maintain constant surveillance. Autonomy, in other words, as an unintended consequence of epidemiology. That autonomous time became the protected space where the culture survived. Language spoken without European ears present. Rituals maintained without European interference. Rice cultivation techniques, basket weaving patterns, musical structures, spiritual traditions preserved inside the gap between exploitation and geography. The culture was not preserved because anyone permitted it. It was preserved because the swamps were lethal to the people who wanted to destroy it. What the genome recorded in this period is astonishing in its specificity. Two men named in the Anson Street burial register, buried within feet of each other, were genetically more distinct from each other than either was from living populations in three different West African countries. They did not share an ethnic identity. They shared a condition. Slavery had invented a category, African, that the genome consistently refused to recognize. Every body buried in that parking lot was from somewhere specific. The cargo manifest had erased the specifics. The DNA had not. University of Connecticut, 2022. Dr. Raquel Flesquez is holding a fragment of bone from the Anson Street burial site. Under the right conditions, the right temperature, the right soil chemistry, the right number of centuries, mitochondrial DNA can survive long after everything else has decomposed. The maternal lineage, passed from mother to child without interruption, without recombination, without alteration, can outlast the body by hundreds of years. She extracted it from 31 individuals. Every single one carries an African haplogroup. Haplogroups L0 through L3, the oldest, deepest branches of the entire human maternal tree, they originate in sub-Saharan Africa. They are the root from which every other haplogroup on Earth eventually diverged. When you find them intact in a burial beneath a parking lot in South Carolina, you are looking at an unbroken maternal line that runs from the coast of West Africa to that specific ground, without interruption across 2.5 centuries. But the individual results are where this becomes precise in a way that history never was. One individual in the Anson Street sample carried an H1CB1A haplotype, a lineage found specifically in Fulani populations of West Africa, not West Africa as a region. One community, one origin, retrievable after 250 years. Another individual carried a Native American A2 mitochondrial haplogroup, a traceable ancestral crossing between an enslaved African and an indigenous person in 18th century Charleston. A life the historical record never documented. A lineage the genome refused to erase. In a 2025 study from Howard University, researchers Jennifer Caldwell and Imani Sykes sequenced mitochondrial DNA from 79 Gullah Geechee individuals and compared them to 110 African Americans from other regions of the United States. In the living Gullah Geechee population today, L3 haplogroups appeared at a frequency of 54.7%, L2 at 28%. Non-L haplogroups, the European and Native American maternal lineages that show up in the broader African American population, were present in fewer than 2% of Gullah Geechee samples. The Sea Islands did not just preserve a culture, They preserved a genome at a level of integrity that nowhere else on this continent can match. And then the database showed something that no one was prepared for. The Ely-Jackson database, a reference library of over 4,000 African mitochondrial DNA variants built specifically to help African Americans trace ancestral origins, could not match more than 30% of the Gullah Geechee haplogroups it was given. These were unambiguously African lineages, clearly part of the L megahaplogroup, but they did not match any population in any reference panel built from any surviving African community on Earth. The people who carried these haplogroups came from communities so geographically specific, so thoroughly dismantled during the slave trade, that the 21st century still cannot name them. The genome survived. The population it came from did not. That gap in the database is not a technical failure. It is the genomic record of which African communities were destroyed so completely that they left no reference population for science to compare against. The absence in the data maps onto the violence in the history with mathematical precision. You can measure the erasure in unmatched sequences. His name is Daba. That is what the researchers called him, one of only 2 names found anywhere in the Anson Street burial record. His bones carry a specific story that the genome can now read with a precision history never allowed. The strontium isotope ratio in his skeletal remains, a chemical signature that reflects the precise geology of the region where a person spent their childhood absorbed through food and water from that specific landscape, indicates Daba was born in West Africa, not in America. He was transported to Charleston as an adult. He did not grow up in slavery. He was taken into it. His Y chromosome is entirely African. His mitochondrial DNA is entirely African. He died in Charleston in the second half of the 18th century and was buried without ceremony, without a record that preserved his full name, without anyone permitted to grieve him publicly. In December 2024, a team publishing in Nature Communications mapped the strontium isotope landscape of sub-Saharan Africa using 266 environmental samples from 24 African countries. It is now the most precise geolocation instrument ever built for tracing enslaved individuals to specific African regions, narrowing origin from a continent to a geological province to a specific river basin to specific ground. Daba's bones carry the chemistry of a landscape that science can now point to on a map. What science cannot recover is how long the crossing took, what he saw, what he called himself before Charleston assigned him a cargo entry. Now hold this for a moment, because what Zimmerman's 2021 study found in the living Gullah populations carries information that cannot stay abstract. The European ancestry in the Gullah genome arrives almost entirely through the paternal line, the Y chromosome. When the researchers analyzed the admixture pattern, comparing autosomal DNA, mitochondrial DNA, and Y chromosomes simultaneously, the evidence for sex-biased gene flow was unambiguous. European ancestry entered through European men. The mitochondrial DNA, the maternal lineage, remained African. The language of the published paper is precise and deliberate. The pattern is consistent with increased frequency of sexual interactions between European men and African women, including, in the researchers' own words, rape and coerced sexual interaction. Virtually every European X chromosome in the Gullah genome came from men. Almost no European women produced children within the Gullah community. The maternal line held. The paternal line was taken. Daba's Y chromosome is intact, African. The admixture that appears elsewhere in the Gullah genome did not reach him. He died before it reached the women around him. He was buried next to men born in America, men whose Y chromosomes told a different story, carrying lineages that crossed the ocean a generation earlier inside the bodies of the men who transported them. The chromosome is not a metaphor. It is a record, and it is in living Gullah men today. The health consequences are also in living Gullah bodies today. Researchers studying the Gullah Geechee community have documented elevated rates of hypertension, systemic lupus erythematosus, and end-stage renal disease significantly higher than in non-Gullah African Americans. Whether this reflects genetic susceptibility specific to certain West African populations, the physiological consequences of multi-generational trauma, concentrated environmental exposure, or some interaction among all 3 remains under active scientific investigation. The genome may not just be carrying ancestry. It may be carrying the body's own account of what that ancestry costs to preserve. Medical University of South Carolina, Charleston, 2004. Researchers studying lupus prevalence in African Americans need a clean comparison population, one with high African ancestry and minimal European admixture, where the genetic signal won't be obscured by centuries of mixing events. They choose the Gullah Geechee, not because of their history, not because of what they represent culturally, because they are genomically useful. That sentence deserves to sit on its own for a moment. The Gullah Geechee have been a medical research population for decades. The Regard Study, the Jackson Heart Study, multiple lupus genetics consortia. Study after study used Gullah samples specifically because their genetic isolation made them ideal research controls. And in that same period, the Sea Island communities providing those samples lost significant portions of their ancestral land to development, received inadequate infrastructure investment, and watched their young people leave for opportunities that did not exist on the islands. The structure of that research relationship is worth naming clearly. The community provided the biological material. The institution produced the papers. The land continued to be sold. Dr. Diane Kamen and Dr. Gary Gilkison at the Medical University of South Carolina have conducted rigorous, critical work on lupus genetics within the Gullah cohort, work that genuinely matters scientifically. The issue is not the science. The issue is what the science was built on without acknowledging it. Then one person changed the structure. Queen Quet, chiefess and head of state of the Gullah Geechee Nation, based at St. Helena Island, is listed as a co-author on the Zimmerman 2021 study in the American Journal of Physical Anthropology. She is not a geneticist. She is listed because she insisted the community would be a partner in research about itself, not merely a source of blood draws and cheek swabs. That insistence is itself a data point, not about genetics, about what every previous study had failed to do, about what research without that insistence looks like, and what it produces, and who benefits from it. There is one analysis that has not been run. No research protocol has modeled the genomic consequences of the community's ongoing displacement. What happens to haplogroup frequencies, to the purity of maternal lineages, to the genomic signal that makes the Gullah uniquely valuable as a research population? When the geographic isolation that produced that signal is dismantled, the data to run that analysis exists. The question has not been formally asked. It is about to need to be. Howard University, Washington, D.C. 2025. Dr. Jennifer Caldwell and her team had been doing something no previous study attempted. They were not simply comparing Gullah Geechee haplogroups to West African populations. They were comparing them to the broader legacy African American population, African Americans from across the entire North American continent, looking for a specific pattern, and they found it. Hold everything I have told you so far: the parking lot, the malaria, the Sierra Leone myth, the violence encoded in the Y chromosome, the health data that no one has fully resolved. Hold all of it simultaneously, because it is about to mean something different. The haplogroup distributions between Gullah Geechee individuals and non-Gullah African Americans were not significantly different from each other. L2 and L3 dominated both populations. Non-L lineages, the European and Native American maternal markers, appeared in under 2% of Gullah Geechee, and measurably higher in non-Gullah African Americans. From that pattern, Caldwell and Sykes made a proposal that the field is still processing. The Gullah Geechee maternal lineages may represent the founding biocultural progenitor population for legacy African Americans across all of North America. Read that again carefully. The most intact African genome in the Western Hemisphere is not a regional variant of the African American population. It may be the biological source of it. Here is the mechanism the researchers propose. The Gullah Geechee remained genomically isolated for generations while the rest of the African American population moved. Between 1910 and 1970, roughly 6 million African Americans relocated from the rural South to northern and midwestern cities in the event historians call the Great Migration. They carried lineages that across subsequent generations diversified through new admixture events in new places. The people who stayed, who remained on the Sea Islands inside the geographic isolation that malaria had originally created, held the source code. The Great Migration was not just a demographic event, it was a genomic dispersal event, and the Gullah Geechee were the point from which the dispersal occurred. You do not have to be Gullah to carry this lineage. If you are of African American descent anywhere in North America, the probability that your maternal line passes through the Sea Islands at some point is not symbolic, it is statistical, and now this becomes a problem of a very specific kind. The most complete biological record of pre-slavery West and Central African maternal diversity existing anywhere in the Western Hemisphere belongs to a community of roughly 500,000 people living on barrier islands and coastal lowlands that are currently being consumed by luxury real estate, development, and rising sea levels. It has no legal protection as a genomic heritage site. It does not appear on any endangered registry. The genome that survived 300 years of slavery, epidemic disease, Reconstruction's betrayal, and the dispersal of millions is now being systematically erased by property law. a genetic lineage intact enough that researchers in 2025 can propose it as the maternal origin of an entire nation's diaspora, living, breathing, in people who are currently being priced off their ancestral land and protected by no statute anywhere in the United States. To understand how the land was lost, you need to understand how it was promised. St. Helena Island, South Carolina, January 1865, General William T. Sherman issues Special Field Orders Number 15. The Sea Islands and a 30-mile coastal strip stretching from Charleston south into Florida are to be redistributed to formerly enslaved families in 40-acre plots. The Gullah, who had already been farming these islands largely autonomously during the Civil War, who had already demonstrated, during years of Union occupation, that they could organize, govern, and sustain themselves without European oversight, believe the land is finally, legally theirs. In a real sense, it already was. Built by their knowledge, fertilized by generations of their labor, defended from disease by the immunity in their blood. 8 months later, President Andrew Johnson revoked the order. The land was returned to former Confederate owners. Gullah families who had already received official land titles were displaced, some violently. By 1870, through the Freedmen's Bureau and private purchase, Gullah families had reacquired approximately 10,000 acres on St. Helena Island alone, bought with wages from agricultural labor, penny by penny, deed by deed, legal instruments purchased at significant personal cost, representing the deliberate reconstruction of what had been taken. Then the 20th century arrived with a different set of instruments. Here's property. Land passed through generations without formal wills, common in communities with historically limited access to legal services, carries no clear title under American property law. It can be seized for unpaid property taxes. It can be sold at auction without the consent of all heirs. It can be purchased by developers who understand its legal vulnerability better than the families who have lived on it for 5 generations. By 2020, Gullah land ownership on the Sea Islands had contracted dramatically from its Reconstruction-era peak. The legal mechanism was different from 1865. The outcome was identical, and now there is a third force operating simultaneously. The IPCC projects 1 to 2 feet of sea level rise along the South Carolina coast by 2050. Gullah communities on Sapelo Island, Dofusky Island, and the St. Helena lowlands are already experiencing tidal flooding on land their families have held since Reconstruction. The barrier islands that malaria made impenetrable are going underwater on a measurable timeline with a tide table attached to it. Development pressure from resort construction. Here's property vulnerability and the tide. Emery Campbell, former director of the Penn Center on St. Helena Island, a Gullah man who watched Hilton Head transform from a majority Gullah community to an international resort destination in a single generation, described watching the landscape of his childhood become architecturally unrecognizable. The golf courses were named after the natural features they replaced. One resort retained the word plantation in its name. The name survived. The people who built it did not. The legal instrument that could protect Gullah Geechee land, and the genomic isolation that makes their DNA uniquely significant to science, to medicine, and to the biological history of an entire people, does not exist. The Gullah Geechee Cultural Heritage Corridor Act, passed in 2006, provides congressional recognition, a corridor designation, a website, a logo. It cannot prevent the sale of a single acre. It contains no land protection mechanism of any kind. The genome is being lost through property law that does not know it exists. The same legal apparatus that stripped the land in 1865 is doing so now through different paperwork. The war did not end. It incorporated. If your maternal ancestry is African American, anywhere in North America, not only in South Carolina, not only in the South, there is a statistically meaningful probability that somewhere in your mitochondrial lineage, a woman stood on one of these islands. She may have carried rice seed across the Atlantic in the only place it could survive the crossing. She may have planted it in the same tidal swamp that killed the overseers who claimed to own her. She may have spoken a language that is still spoken today in Sierra Leone, even though her genome tells a more layered story than that single connection allows. She did not know she was preserving something that would be sequenced in a Connecticut laboratory 250 years after her death. She was surviving. The survival happened to be genomic. What the science now shows is that the survival was also foundational. The Great Migrations dispersed her lineage across an entire continent. Every city that received those migrants in the 20th century received the lineage she had kept intact on an island surrounded by malaria. The haplogroup frequencies that Howard University measured in Gullah Geechee individuals in 2025 are the source frequencies, the predispersal baseline for a population that now numbers in the tens of millions. The 36 people beneath the Anson Street parking lot are reinterred now. Charleston built a memorial fountain above them, a water feature over the bones of people the city processed as cargo and buried without ceremony. The water runs continuously. It was not named after any of them. The Gullah Geechee Cultural Heritage Corridor stretches 425 miles from Wilmington, North Carolina, to Jacksonville, Florida. It has a website, a congressional designation, a logo. It does not have a legal mechanism to prevent the sale of a single acre of the land that made the genome possible. The most intact African maternal lineage in the Western Hemisphere has a corridor. It does not have a deed. And the tide table on the South Carolina coast does not consult congressional designations before it moves.